we are going to talking about the structures of different polymerases which plays a role in the replication. And the first enzyme, which was identified by a scientist, Arthur Kornberg, and he named this enzyme as a Kornberg enzyme, and later it is named as a DNA polymerase 1. And it is not essential for actually the DNA replication process, but it helps in the repair replication, and it has some sites by which it actually helps in the repairing process. And it is also called the metalloenzyme because it has a single polypeptide chain, and therefore it is called the metalloenzyme. It has actually two fragments. One is called the Kleno fragment and another is called the smaller fragment. And electron microscopic studies showed that it has a number of sites which plays an important role in replication process, and each site has its own role which is different from each other. The first site, which is called the template site, and this template site actually helps to hold the strand of the DNA. Therefore, we called it as a template strand. And we know that for the DNA replication process, it always requires a primer of RNA molecule, which actually complementary to the template strand. And to holding this primer stand, this enzyme also has another site, which is called the primer site, and where the primer actually fits to the polymerase, and it actually complements the template site, and it forms a hybrid at this situation. And just upper to the position of primer site, we has another site which is called as a triphosphate site, where an incoming nucleotide, it's like maybe a ATP, or it's maybe a GTP, or maybe UTP. This triphosphate molecules are actually comes to this site, and it binds with the primer at the 3' OH position and matches with the complementary base on the template strand. And another two essential sites of this polymerase, one is called a 5' to 3' exonuclease site, another is called 3' to 5' exonuclease site. And I think you already know about what is the exonuclease activity. All the DNA polymerase has some kind of exonuclease activity which helps in removing some faulted bases or some misincorporated bases of DNA during the repairing process. And why these are called exonucleases? Because this helps to remove the bases from each end of the DNA, not from the middle position. If some polymerase helps to remove the bases in middle position of the DNA, in this situation we called it as a endonuclease. But a function of exonuclease is actually it removes the DNA from a free end. So it has both kind of activity. It has 5' to 3' exonuclease activity. That means it removes the bases from 5' to 3' direction, as well as it has also 3' to 5' exonuclease activity, by which it removes the bases from the 3' to 5' direction. So this is all about the DNA polymerase 1, and in our next video we are going to talking about the other polymerases which helps into the DNA replication process.